Bismillah ar-Rahman ar-Rahim. My name is Dr. Khalid Abul Bashar. Uh, I welcome you all to my YouTube channel, the channel that I call Molecular Genetics. Uh, this lecture is Cell Cycle Part 2. In Part 1, I have talked about cell cycle, an introductory part, and I have discussed three phases of cell cycle, G1, S phase, and G2, and their means of regulation. In this lecture, I will talk about the final phase in the mitosis, final phase of the cell cycle, which is mitosis. So, what is mitosis? Mitosis is the final conclusive step in a single round of cell cycle, and it is normally consists of four phases. But in some books, you find five phases. The four phases are Prophase, Metaphase, Anaphase, and Telophase. In some books and references, you find an additional phase that is Pro-Metaphase, which is a phase between Prophase and Metaphase. There is no clear-cut line that separates these phases. Uh, each phase is characterized by distinct cellular activity, and this cellular activity they are the determinants of each phase. So we rely mainly on the activities and reorganizations that takes place inside the cell to determine at what phase the cell is. Mitosis starts by a number of changes. The cell is subjected to a number of changes in the mitosis, and it is actually a step where the cell is completely uh, divides into two to form two daughter cells, mostly genetically identical daughter cells. So the whole process, the aim of the whole process is to give two daughter cells from a single cell. So I find so many activities that take place inside the cell in preparation for the cell to divide finally into two daughter cells. Number of changes that take place inside the cell lead to what is known as karyokinesis. Karyokinesis is the division of the nucleus. The nucleus at one step in the mitosis divides into two nuclei, genetically identical nuclei, and the cell continue to reorganize its internal environment and prepare itself for the final step, which is the cytokinesis. Cytokinesis is the final step, which is a complete separation, the split of the cell into two. So the mitosis start with karyokinesis, which is the division of the nucleus of a cell into two nuclei, and then the cell continue doing some work, preparing itself for the final step, which is the cytokinesis. That is a complete cell division. The cell will divide into two. Now we talk about each phase independently and in more details. The first phase in the cell in the mitosis is prophase. Prophase is the first phase in the cell, and it is a preparatory phase that the cell prepare in which the cell prepare itself for a complete nucleus division. In this phase, we find so many changes that take place inside the cell. One of the main change that happens during the prophase is the DNA condensation. So DNA condensation is a very important step in the way of cell to divide itself into two. So why DNA condenses? Normally in a cell we find a DNA in one of two conditions. Either the DNA is wrapped tightly around its histone protein to form what is known as chromosomes, distinct structures known as chromosomes, or sometimes sister chromatids when we talk about it from a cell cycle perspective. And another state or another condition in which the DNA is found loosely bound to its DNA to its histone protein. So the DNA either tightly bound to its histone protein, forming a distinct structures known as chromosomes, or it is loosely bound around its histone protein, and at this point, the DNA is known, or the structure, this structure is known as chromatin. So the DNA is either DNA and its histone protein, either chromosomes or chromatin. The chromosomes are not formed unless the cell is dividing. The only the only stage at which the DNA, the genetic material of the cell, is found in the chromosome, these distinct structures, is during the cell division, during mitosis. During the interphase, when the cell is not dividing, we find that the DNA is the genetic material is rather found in a loosely bound structures known as chromatin. So why the DNA condenses during the prophase? The DNA condenses and wrapped itself around the histone protein tightly, so that it makes a distinct structure. This structure is smaller, stronger, and easy to separate during the separation of the chromosomes. We have to bear in mind that the DNA in prophase is actually coming from the S phase. The cell has passed this step first, and then it went to the S phase, where the DNA has been replicated. Each chromosomes in the S phase now has been replicated, and we find for each chromosome there is two copies. So these copies are still attached to each other at certain points. The DNA of the two copies of the chromosomes are attached to each other at certain points. And then the cell will pass through this G2 and then goes to the prophase. In this prophase, the DNA, the two copies of the chromosome are actually attached to each other, and they start condensing, forming what is known as a chromosome, or a more specifically known as sister chromatid. So the DNA will condense to give a smaller structure, more smaller structure, stronger structures, and easy to separate structures, so that it can be so easy for the cell during the later phases of the mitosis to separate these structure away from each other. And this structure is also less susceptible to breakage during the movement across the cell during the different phases of mitosis. And it's worth mentioning that DNA at this point is transcriptionally inactive. So gene expression at its lowest rate. The DNA is different genes in these structures, chromosomes or chromatin or whatever it is. They are at this point they are not expressed at all. So this is the first change that takes place during the prophase. The other change is the breaking apart of Golgi bodies, Golgi apparatus, and the endoplasmic reticulum. These structures normally break apart into membranous vesicles that can be easily and evenly distributed across the cell for an even distribution during or after the cell is divided. So the Golgi apparatus, these membranous organelles, Golgi apparatus and endoplasmic reticulum, are broken apart so that it can be easily uh, divided during the cell division. Another thing that happens during the prophase of the mitosis, that the centrosomes. Centrosomes normally are found near the nucleus. When the cell enters the prophase, centrosomes start moving away from each other toward the opposite sides of the cells to establish what is known as the poles of mitotic spindles. So these centrosomes starts moving away from each other to the opposite sides, forming what is later to be known as the poles of mitotic spindles. This is also a very important step that takes place during the prophase. Another very important and vital step, that is the action of the MPF. MPF is known as the maturation promoting factor. Maturation promoting factor, and maturation promoting factor is actually a cyclin dependent kinase complex. It is a complex that is a cyclin dependent kinase complex, and it phosphorylates a number of proteins. Some proteins known as MAP or MAPs. These are microtubules associated proteins. And the MPF also phosphorylates microtubule motor proteins. So in doing so, in phosphorylating the motor protein of the microtubule motor protein and the MAP protein, this is actually alter the normal microtubule dynamics. So the phosphorylation and the activation of these proteins will alter the dynamics of the microtubule, and now the microtubules, these are small structures, proteinous structures, filamentous structures coming out from the centrosomes, and these structures now, after the phosphorylation by the MPF, they will become more dynamic and they will reorganize easily so that they can form what is later to be known as the mitotic spindles. So this is very important step. One of the protein, an example of this is a protein known as PRC1. PRC1 is also known as, this is a short form of a protein regulator of cytokinesis 1. Protein regulator of cytokinesis 1. This is a bundling protein. It's a protein that keeps the microtubules together as a bundle, and it's normally gets inactivated by phosphorylation. So by the action of the MPF, this PRC1 will get inactivated, so they release the mitotic proteins, these microtubules. Now these microtubules are more free, and they can freely move away from each other and they can easily move to new location. So all this will increase the dynamics of the microtubules by releasing the PRC1. This is just an example of what happens. The PRC1 will release the microtubules and make them easy to relocate themselves and reorganize themselves so that they can form what is to be known as the mitotic spindles. In the early stage of prophase, we find that the DNA of the two copies of the chromosomes um, resulted from the S phase. These two copies of the DNA of the chromosomes, now they will condense and will wrap themselves around each other, around the histone proteins, forming what is to be known as sister chromatid, and then the breaking apart of Golgi bodies, Golgi apparatus, and the ER, and then the movement of the centrosomes away from each other, forming the what is later to be known as the poles of the mitotic spindles, and with the action of the MPF, the maturation promoting factor, which is a cyclin dependent kinase complex, a number of protein gets activated and inactivated, helping in an easy and increasing the normal dynamics of the mitotic spindles or the microtubules, so they can be easily organized and relocated during a later stage. In the prometaphase, or what is also known as late prophase, it's either referred to as a prometaphase or a late prophase, the disintegration of the nuclear envelope takes place. This happens by the help or by the action of the MPF. The protein MPF, as I said, it is it's a cyclin dependent kinase complex. This MPF protein, maturation promoting factor, will phosphorylate the lamines protein. If we just go back to the structure of the nucleus, we find that the nuclear envelope, under the nuclear envelope, there is a cytoskeletal structure known as the nuclear lamina, which give the support to the nuclear envelope. So the nuclear envelope normally attach itself to a nuclear lamina, and the nuclear lamina is made of the lamine protein, and the phosphorylation of these these lamine proteins by the MPF will make these lamine proteins rich with the phosphate group. And chemically, we know that the phosphate group are carrying negative charge. So now the lamines are all covered with a negative charge due to the presence of the phosphate. From where? From the action of the MPF, phosphorylation by the MPF. So the lamines, the building units of the nuclear laminae, are now covered with a phosphate group. Phosphate group, as I said, they are negatively charged, and this negative charge will make these lamines more repulsive to each other. They will change the nature of interaction between the lamines from being closed and attached to each other. Now the lamines are more repulsive to each other due to the presence of the negative charge, and they will move, dissociate away from each other, uh, uh, breaking down the structure, the nuclear lamina, and the nuclear lamina to which the nuclear envelope is attached. This will result in the disintegration, dissociation of the nuclear envelope, and the disappearance of the nuclear envelope and disappearance of the nucleus in its uh, 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 normal structure. So with the help of the MPF, now the nuclear envelope will disappear, disintegrate, break up, and this happens due to the phosphorylation of the lamine protein, the building blocks of the nuclear lamina. Now, the disappearance of the nuclear envelope will make it quite easy for the mitotic spindles to attach to the chromosomes. Now, as I said, as I said earlier, that the centrosomes will move away from each other, forming what is later to be known as the poles of mitotic spindles. The mitotic spindles, these structures will try to attach themselves to the chromosomes. So the mitotic spindles at this point, they will increase in size and decrease in size. They increase their dynamics quite rapidly, and they keep on polymerizing, and they will undergo polymerization and what is known as a catastrophic disassembly. So they will increase in the length, and then they will decrease in the length, trying or searching for a chromosome to attach themselves to. So these microtubules will keep on searching for a chromosomes to attach after the disappearance of the nuclear envelope. Now the spindles, the microtubules, these microtubules will keep on increasing in length, decreasing in length, increasing and decreasing through polymerization and catastrophic disassembly. searching for a chromosome to bind to. So there are three types normally of microtubules. These are the microtubules. We normally have three types of microtubules, or this microtubules of the mitotic spindles. This is all known as the mitotic spindles, and we have three types of microtubules. We have one type of microtubule called as, or known as, Kaintucore microtubules. Kaintucore microtubule, Kaintucore microtubule, and this Kaintucore microtubule is a type of a microtubule that extends from the centrosomes, and it attaches itself to a point, a proteinous structure in the chromosomes known as the Kaintucore. So the microtubule, Kaintucore microtubule, will bind itself to the Kaintucore. This is from where the name has originated. The microtubule that binds to a Kaintucore is known as the Kaintucore microtubule. So this is, and it's very important. It has a pivotal role in the separation of the chromosomes, uh, separation of the chromosomes in a later stage. So this is the first type. The second type of microtubule is known as a polar microtubule, and polar microtubule actually reaches across the cell and interact with one another. So this will extend all the way, and the other one coming from here will extend all the way until they attach to each other, and they have many roles in the separation of the chromosomes. They help in maintaining a separation of centrosomes, so they keep these centrosomes away from each other. They help maintaining these centrosomes separated, and they also define the overall length of the mitotic spindles. So the polar, and we will later see how they help in the separation, the process of chromosomal separation during the anaphase. So the second type of the microtubule is the polar microtubule, which reach across the cell and attach to the other one coming from this side, and they help in maintaining these centrosomes away from each other, and they help later on in the separation of the chromosomes. The final type of microtubule is the aster microtubule. This is polar microtubule, Kaintucore microtubule, and the aster microtubule. Aster microtubule are actually short, and they radiate and stabilizes centrosomes, and they help in the separation of chromosomes during the anaphase. So also these aster, they are short microtubule, and they radiate from the centrosomes, and they help in the separation of the chromosomes later during the anaphase. So they have a role to play during the separation of the chromosomes. They are very important for this process as well. So now we have talked about the microtubule and the three types of the microtubules. Let's talk a little bit about the Kaintucore. We know that in the S phase, as I said, the DNA has been replicated, and each chromosomes now we find two copies of that chromosome. We have a two copy from each chromosome. This is one. This is one copy of the chromosome. The other one is the other copy of the chromosome. So this is a chromosome, and this is a chromosome. These two chromosomes normally in the S phase, they do not separate completely. Rather, they they get attached at certain points. The DNA of these two copies gets attached at certain points, and there is a point at which the DNA and the histone protein they have a very strong attachment, and this very tight wrapping of the DNA around the histone protein is due to a presence of repetitive sequences in the DNA. So the presence of repetitive sequences in the DNA makes the DNA more prone to a stronger and tighter wrapping around its histone protein, giving this constricted region in the chromosome is known as centromere. So this centromere region is due to the presence of certain repetitive sequences in the DNA. The presence of these sequences make the DNA bind stronger to the histone protein, unlike other regions. So the DNA here binds stronger with the histone protein, giving this tighter, more constricted region in the chromosome, and this region is known as a centromere. The centromere is there is a proteinous structure that is bound to a centromere from this side and the centromere from this side. As I said, this is a chromosome and this is a chromosome. The whole structure, the two chromosomes, are known as sister chromatid. Sister chromatid. Why sister? Because they are two copies from the same chromosome. So in the S phase, the chromosome has been copied into two. So these are the two copies of the same chromosome, and they are known as sister chromatid. They are held together in the region of centromere, and in the outer side of each centromere, there is a proteinous structure known as kinetochore. So, what is the structure of the kinetochore? The kinetochore is a proteinous structure. It is made of three subunits: the outer subunit and the inner layer, the outer layer, the inner layer. They have very important role in attaching the whole structure to the centromere region. And we have what is known as the fibrous corona. Fibrous corona, these are filamentous proteinous structures. They are radiating. They normally radiate from the outer structure of the outer layer of the kinetochore. So we have three structures: the inner layer, kinetochore layer, outer kinetochore layer, and then we have a fibrous corona. Normally, the kinetochore microtubule, this is a kinetochore microtubule, the microtubule that binds to the kinetochore in the constricted region in the centrosome. This microtubule normally binds to the fibrous corona. It does not reach all the way until the outer layer or the inner layer. Instead, it binds to the fibrous corona through a number of proteins. One of the protein that helps in binding, in attaching the microtubule, the kinetochore microtubule, to the fibrous corona of the kinetochore is the CENP or centromere binding protein E. Centromere binding protein E. This is a protein, and we have also dynein protein. This motor protein also help in binding the microtubule, the kinetochore microtubule, to the fibrous corona. So the CENBE and kinesin, dynein, these are motor protein which help in binding the fibrous protein to the kinetochore microtubules. after the kinetochore microtubule gets attached to the kinetochore, they keep on decreasing and increasing in size. They keep on decreasing and increasing in size. And now the chromosome, this sister chromatid structure, this chromosomal structure, the whole thing is bound by two kinetochore microtubules. One coming from this side, from this centrosome, the other one coming from this side. Each microtubule subject a certain level of tension. So the structure, the chromosomal structure, is subjected to certain level of tension from this side and certain level of tension from the other side. So the structure, the chromosomal structure, will keep on fluctuating from this side to other side until the level of tension that is subjected by this part, by this kinetochore microtubule, equals that is coming from this side. Until the level of tension is equal between the two sides, this structure keeps on moving from side to side. So when when the tension applied by this microtubule equals that tension applied by this microtubule, the cell will recognize that the chromosomal structure has come to the middle of the cell. So the main aim of the movement of the kinetochore, this continuous dynamic movement of the kinetochore structure, the microtubule kinetochore microtubule structures from both sides, is to align the chromosomes in the middle of the cell. So how the cell knows that all the chromosomes are now aligned in the middle, or what is known as the mitotic plate or the equatorial plate of the cell, the midline of the cell. How the cell would know this? The cell would know this when the tension applied by this side equals the tension applied by this side. But if the chromosome is in this side, in this area, for example, at this point, this chromosome is bound, and this microtubule, this kinetochore microtubule, goes all the way till this point. So what happens? That means the tension applied, the pulling tension, the tension applied by uh, uh, microtubule is less than that applied by the whole thing, by this whole microtubule that is coming until this point. So that means when one side applies more tension than the other side, that means the chromosome has not yet reached the midline of the cell. Until the tension is equal between both sides, and the chromosome keeps moving from this side to this side until it reaches the midpoint. So now, when all chromosomes have reached the midline of the cell, now the cell is said to be in the metaphase. So the metaphase is characterized by the alignment of the cell in the midline or in the center of the cell, the alignment of the chromosomes in the center of the cell. This will indicate that the cell has reached the metaphase. So sister chromatid, or this chromosome or sister chromatids, are lined up along the midline of the mitotic spindles at this point. Now the cell is said to be in the metaphase. This metaphase is rather a relatively static phase. There is no reorganization or internal activities take place in this phase as what happens in the earlier or the later phases. So it is a relatively static phase. Here the MPF also, the cyclin dependent kinase complex, MPF, gets inactivated, and this is done partly by its own action. How? This MPF actually at the metaphase, it will phosphorylate and activate a protein called APC. This normally happens at very late at the metaphase. So late at metaphase, at the late stage of the metaphase, the MPF will phosphorylate the APC. APC is anaphase promoting complex. Anaphase promoting complex gets activated by MPF. MPF, it's a maturation promoting factor. So it activates this protein, and this protein in turn polyubiquitinate the MPF. It adds number of ubiquitins to the MPF. So ubiquitination of the MPF will indicate that this protein will be subjected to destruction. We know that ubiquitination means ubiquitination of protein is a signal for the proteasomes to destroy that protein. So the MPF will activate the APC, and APC in turn will polyubiquitinate the MPF. Now MPF will be destructed. It's rather the cyclin part of the MPF. As I said, it is a cyclin dependent kinase. So the cyclin subunit of the MPF will be destructed, and so the MPF will be inactivated. So this happens during the late stage of the metaphase. APC also tagged. It also add ubiquitin to a protein called securin. Securin also, it's a very important protein, securin. Securin is a protein that binds and inhibits the action of an enzyme called cyprase. So the securin normally binds and inhibits the action of an enzyme called cyprase. Cyprase is a very important enzyme for the metaphase, for the mitosis. So APC will first help in the destruction of the MPF, and APC will also tag the securin, so it will be destroyed. So MPF will be inactivated and securin will be subjected to destruction by the action of APC. So it's also at this point the chromosomes. It's worth noting that chromosomes at this stage, the metaphase, chromosomes are highly condensed. Though they have not reached the peak of its condensation, but at this point the chromosomes are highly condensed. And this chromosome, the metaphase chromosomes, are chromosomes that can be easily seen during the easily seen under light microscope. And normally when we study chromosomes at this stage. So the metaphase chromosomes are normally the chromosomes that are used to study chromosomes for karyotyping and studying the cytogenetics and this. So at this point and in this stage, the chromosomes are highly condensed and can be easily visible and seen by light microscope. Sorry, excuse me. After that, the cell will pass from metaphase to anaphase. Now the cell will reach anaphase after the destruction of securin. So destruction of securin means that the cell has reached the destruction of securin and activation of anaphase promoting complex. Normally, as I said, that sister chromatids are held together at a region by a protein. So sister chromatids, as we have seen earlier, sister chromatids are held together at this point. They're held by a protein known as cohesin. So cohesin protein help in the attachment of both sister chromatid to each other, both chromosomes to each other, forming a sister chromatid. So destruction of securin will activate a separase enzyme. Separase enzyme, we said that securin normally binds to separase enzyme and inactivate the separase enzyme. Now securin has been destroyed with the help of the APC. Securin is destroyed, and destruction of securin leads to the activation of separase enzyme. Separase enzyme will go and destroy and break down the cohesin protein. Cohesin protein, the protein that help in holding the two chromosomes together to form sister chromatid. Now sister chromatid are separated. They are free from each other. The sister chromatid, now they are freely to move away from each other. Anaphase is also divided into two phases: early anaphase and late anaphase, sometimes called A and B. Early anaphase. In early anaphase, kinetochore microtubules, these microtubules, kinetochore microtubules, which are bound to the kinetochore, will shortening. They start to get shorter and shorter by time from both ends, from this side and from this side. So they are shortening in length and pulling away the chromosomes away from each other. The shortening of the kinetochore microtubule will lead to separation of chromosomes away from each other. This is in early anaphase. In late anaphase, the polar microtubules, polar. Now, the action of the kinetochore microtubule is by shortening themselves and getting separated away from each other, pulling away the chromosomes. Now, in late anaphase, the polar microtubules, these microtubules, they are still attached to each other by the head. Head of this microtubule is touching the head of the other microtubule. So this polar microtubule will elongate. They will increase in size, elongate, and additional chromatid separating force is applied by kinesin 5 motor protein that push the polar microtubule against one another. So they will elongate the polar microtubule, and this will elongate, and they touch each other with the help of a motor protein called kinesin 5. A kinesin 5 protein will push the polar microtubule against one another. So each one will be pushing the other, and so what will happen? This will increase the separation between the two poles. So this will push this one, and this polar microtubule will push the other one. So this will help in pushing the centrosomes away, further away from each other, and help in the separation of chromosomes. So an additional chromatid separating force is applied due to the pushing of these polar microtubule pushing one another. So these centrosomes will move away from each other, pulling the kinetochore microtubule to which the chromosomes are attached. And another thing will happen. Now we have seen the role of kinetochore microtubule and the role of the polar microtubule. Now the role of the aster microtubule. To the aster microtubule, a motor protein will bind. This is dynein motor protein will bind to the aster microtubule and direct the binding of this aster microtubule to the actin protein. Normally inside the cell, in the inner part of the cell, in the inner part of the cellular membrane, there is a cytoskeletal structure known as made up of a actin protein. So there is a cytoskeleton actin protein found in the inner part of the cell membrane or the plasma membrane. So the dynein protein will direct the attachment of this aster microtubule to the inner part, the inner part of this plasma membrane, and help the attachment of these aster proteins, aster microtubule to the actin, which is found in the inner part of the cell membrane. So, and these aster proteins, aster microtubule, will pull. They will pull the centrosomes towards the plasma membrane. So they will help pulling the centrosomes towards the plasma membrane. So now we have three forces: shortening of the kinetochore microtubule, pushing of polar microtubule one another, and the action of the aster protein by pulling they get attached to the actin protein, which is found in the inner part of the cell membrane, and they try to pull the centrosomes away, and this one will pull the centrosomes away, so subjecting the chromosomes to further separating forces. So by doing so, the microtubule and the mitotic spindles will help in separation of the chromosomes away from each other. So these chromosomes, they will reach their respective poles. So each chromosome will reach this will take place until all the chromosomes will reach their respective poles. By reaching their respective poles, now the cell is said to be in the telophase. So telophase starts when both sets of chromosomes arrive at their respective poles. When both sets of chromosome arrive at their respective poles, the telophase is said to be started. So after that, what happens in the telophase? We know in the anaphase and late metaphase, the MPF has been inactivated. And the MPF, as we have seen earlier in the Prophase, the MPF was responsible of the phosphorylation of the lamine proteins. And phosphorylation of the lamine proteins was the reason why the nuclear envelope has been disintegrated. So now MPF is out of the picture. So it's not there. It has been inactivated. So no MPF, that means no phosphorylation. And at the same time, phosphatases enzymes, cellular phosphatase enzyme, will remove the phosphate group from lamines. So now no phosphorylation by the MPF, and there is removal of the phosphate groups from the lamines by the phosphatases. So the lamines now will come together, will come again and reform the nuclear lamina. Reforming the nuclear lamina means reforming of the nuclear envelope. So now the nuclear envelope and the nuclear lamina will reform, forming the nucleus. So now we have two separate nuclei. Instead we have started with one nucleus in the prophase, and we carried on until we separated this nucleus into two nuclei, and this is called the karyokinesis. So karyokinesis is the separation of a single nucleus into two nuclei in a single cell. So this is all this takes place in the telophase. Also, a Golgi apparatus and the endoplasmic reticulum will start to reform again. They will reform again Golgi and endoplasmic reticulum. And inside this newly formed nucleus, the chromosome will start to decondense. Now, the reason for condensation is over. Now, the chromosome will decondense, and a chromosome turn from chromosome into chromatin. Now they become a chromatin. Now we end up with a single cell with a large single cell and two complete nuclei, independent, separated on the opposite sides of the cell. Now the karyokinesis is done. Now the cell will go to a step called cytokinesis, or the complete cellular division. How the cytokinesis happens? Cytokinesis actually starts by the formation of what is known as the contractile ring. Contractile ring is a ring formed from actin protein and myosin motor protein. So actin proteins will come together, forming a ring, and with the help of myosin protein, this ring will tighten. It will tighten up, and at the same time, the actin protein, it is actually, as I said, it's part of an inner cytoskeletal structure. So the actin protein partly bound to the inner membrane, the inner plasma membrane, inner cell membrane, and this actin ring is attached to the inner part of the inner side of the plasma membrane. So, and by the action of myosin protein, this ring will tighten up. It will decrease in size in the peripheral. So the ring tighten up, pulling the waste of the cell. So it will decrease in the side and pull the plasma membrane inwards until the edge of the plasma membrane meets, until these edges of the plasma membrane meet. So uh, while doing so, the cleavage, something called as cleavage furrow, is formed. So this cleavage furrow is formed around the cell, and this will happen until the cell is splitted or divided into two. So the contractile ring will keep on decreasing in size with the help of a myosin protein and energy from ATP. This will help in tightening up the ring and pulling the plasma membrane inwards until the cell is divided into two. Normally, the placement of the ring in the middle of the cell, but it is not always the case. We have in a case of a oocyte formation or the oogenesis egg, when as it is very well known that the egg is the major contributor of this cytoplasmic material in the zygote during the zygote formation. So the sperm normally contribute very little of its cytoplasm along with the nucleus in the head, while the majority of the cytoplasm is contributed by the egg. So during the formation of the oocyte, during the oogenesis, the contractile ring is not placed in the middle. It's either placed asymmetrically. One cell will be larger and the other one during the division, this cytokinesis. So the ring will be placed somewhere here in one side asymmetrically. So when the cytokinesis takes place, the cell will divide into two, a very large cell taking the majority of the cytoplasmic material with one nucleus, and another cell taking a scanty amount of the cytoplasm with its nucleus. The larger cell is called the oocyte. It's the oocyte, the egg, and the smaller one is the polar body. So this is an example of an asymmetrical placement of the ring during the cytokinesis. So by doing this, now the cytokinesis has come to an end, and the complete cellular division has took place, and the mitosis is completed. Thank you so much for watching this lecture. I hope that I made things more clear, and looking forward to seeing you in upcoming lectures. Thank you. Goodbye.